And good afternoon, everyone, depending on where you're joining us from, and welcome to today's webinar. My name is Peter Ture from Business Review, and I'll be your host today. It is our pleasure to have with us Ncardia, who will be presenting us on the topic titled On the Road to Discovery, Added Value to Drug Discovery via Human iPSC Disease Models. Now, today's guest speakers of us will be Ari, who is the director when it comes to manufacturing technology, and we have with us Eleanor, who is the director for discovery technology. I'd like to welcome everyone here to On24. You notice that this platform is browser-based. If you do disconnect for any reason whatsoever, please click on the link that you received via email to rejoin the session. In order to ask questions, you can send them in via the questions widget. That's the box at the top right-hand corner of your screen. Just type in your question and click submit. We'll then allocate some time at the end of the session to address any thoughts or questions that you may have. Please also use the yellow help widget in case you need any assistance to move, resize, and maximize any of the screens in front of you to help get a better view of today's session. But for now, please allow me to hand you over to our speakers for the day. Ari, over to you. Yes, Ari, over to you, or you're into Eleanor, over to you. Yes, apologies for that, ladies and gentlemen. We'll just try to find out what's going on at the moment. Let's see. Hello, Alina, Ari, can you hear me? Apologies for that, ladies and gentlemen. We have a little bit of technical difficulties. We'll resolve this as soon as possible. So in the meantime, I'll get in touch with Alina and also Ari, and we'll be able to resolve them shortly. All right, not a problem. Leonie, basically, Alina, she's lost connection. She'll be back. In the meantime, Ari, can you also speak with us? All right, apologies for that, ladies and gentlemen. All right, so, whilst the speakers are getting in touch back in terms of the webinar at the moment, if you have any questions that you would like to submit while our speakers are getting ready, then please feel free to submit them. I'd also like to point you out to the widget bar below you. So for those of you that don't have an idea what the widget bar will be, if you'd like to participate in the survey, feel free to give your feedback on how the webinar will be. We also have some resources from Ncardia that will be very useful for your own download preferences. So if you'd like to, in this case, review over any of the resources, please feel free, as well as submit any of your questions. Our speakers should be with you shortly. All right, the speakers should be rejoining pretty soon. So in the meantime, we'll give it a few more minutes. In the meantime, though, feel free to have a look at the Ncardia resources before we begin this presentation. All right, I think I have got word that both Alina and Ari, they should be joining on shortly. But in the meantime, any questions or thoughts that you have regarding this topic in hand, please feel free to submit your questions and they'll be addressed when it comes to the Q&A session of the webinar. But in the meantime, our speakers will be back on shortly. So thank you very much for your patience, and we would soon be ready to give you a webinar very much soon. Okay. All right, one of our speakers has just joined on, so he'll be with us shortly. In the meantime, we'll wait for him to appear before we start off the presentation. All right, now both speakers have joined on, so both Ari and Alina have reconnected and joined on. So you'll be soon to hear the webinar, Ncardia on the Road to Discovery.
Thank you very much for the kind introduction, and thank you to all of you for joining us today. I will begin this talk by giving the outline of what we will discuss. Initially, we will focus on the background of the drug discovery process. What does it traditionally involve? Then move on to outlining the benefits of iPSC-based disease models and how these can be successfully integrated into a drug discovery panel, and then outline the key considerations or bottlenecks, if you like, that are associated with using this platform for drug discovery. And finally, we will stand on how partnering with experts such as Ncardia can really help overcome these bottlenecks and help you incorporate iPSC platforms into your drug discovery pipelines. So to navigate through this slide, what we can see here is the typical drug discovery funnel, which starts with target identification. And this is the identification of a protein that, when modulated, can really help improve a disease phenotype. This stage is followed by screening of a really large number of compounds, typically over 10,000 compounds, in order to identify hits that can be incorporated or used as therapeutics. And some of these compounds that show favorable profiles are then selected in order to undergo additional studies for efficacy and safety, drug-drug interaction studies, and so on. These are the preclinical stages for drug discovery, and they're followed by long clinical stages before any drug can be approved and be released in the market for use in therapeutic applications. This process is a really long process, and it can take anything up to 15 years for a drug to be developed and be released in the market, and it can cost a large amount of money, up to perhaps $3 billion, which is a very high cost. This process is also associated with a very high rate of attrition, a very high failure rate. And some of this is because the preclinical models that are used for drug development can be suboptimal. They typically include the use of in vitro and animal in vivo studies. So really the use of animal organisms or animal cells that have very little correlation sometimes to the human physiology. Also, human transgenic cell lines can be used, and these can overexpress the target protein of interest, but they can lack the full physiology, the functionality of human cells, such as cardiomyocytes, neurons, and so on. And so this is where iPSCs can help improve the drug discovery process. So to backtrack, iPSCs are induced pluripotent stem cells that are derived from reprogramming of primary human cells isolated from healthy donors or patients carrying genetic mutations. These primary cells are reprogrammed into induced pluripotent stem cells, which can then be also genome edited in order to introduce or correct mutations of interest. And then they can be amenable to differentiation into practically any type of cell in the human body. iPS cells have two very important properties that make them important and useful for drug discovery. One is that they can self-renew practically indefinitely, and this can generate a really large supply of cells that are needed in order to perform a drug discovery campaign. They can also differentiate into any functional type of the human body and therefore really provide a functionally relevant human platform for drug testing. So this makes iPSC derivatives powerful tools that can really retain patient-specific genetic backgrounds, and through thousands of publications to date have been shown to mimic clinically relevant human pathophysiology and to respond appropriately to clinically relevant pharmacology. What this achieves is that we can have a human biological content introduced earlier into the drug discovery funnel so that the success rate of drug discovery can improve, the lead time can be shortened, and the costs can be reduced. So, so this creates really a new paradigm where now into the drug discovery funnels, iPSCs can be incorporated really in every stage. And in the target selection stage, they can be used for target validation in physiologically relevant disease models. In the high throughput screening phase, they can be used in order to perform screening with functional assays rather than non-informative biochemical assays. And in the preclinical development stage, they can be used for successful structure-activity relationship and mechanism of action studies in a, again, clinically relevant human biological context. So as I mentioned, there are some key considerations that are associated with using iPS cells. These include the significant expertise that is required in order to overcome some technical and conceptual challenges. Some of the challenges are associated with sourcing of iPSCs from patients with the right genotype, age, gender, and existence of appropriate clinically relevant match controls. The lengthy process that is associated with consenting patients in order to acquire their primary tissue, and also to select the best tissue to perform reprogramming from, and also the method that will be used to perform reprogramming. Another challenge is associated with the scale-up of differentiation and production of high-quality differentiated cells that have the large enough scale that is needed for high-throughput screening. And this is something that we will stand on during today's discussion. And finally, the assay development associated with iPS cells does require expertise in order to determine the optimal cell density, cell culture media, and coding matrices in order to achieve optimal performance for the cells. And so these are the reasons why partnering with experts such as Ncardia that have spent years developing these technologies can really help overcome the challenges and achieve optimal results in the shortest time frame possible. As a contract research organization, Ncardia really believes that human stem cell biology will help bring better therapies to patients faster. We are a company of approximately 50 employees based in the Netherlands and Belgium, but with business development and marketing presence across the globe. And our services cover the A to Z of iPSC technologies. And this means iPSC sourcing and reprogramming, manufacturing process development and scale-up, differentiation to disease-relevant models, as well as assay development for disease modeling and screening of therapeutic modalities for safety and efficacy, both in large scale and small scale. In terms of our differentiation capabilities, we already have established protocols for high-scale differentiation into cardiovascular and neural lineages. In terms of cardiovascular cells, we can generate ventricular and atrial cardiomyocytes, as well as endothelial and smooth muscle cells, and we will describe some of these in detail in the next slides. In the neural lineages, we have established protocols for differentiation into astrocytes, microglia, cortical neurons, as well as peripheral neurons, and a population of cells that contains both excitatory and inhibitory neurons, as well as glial cells that approximate the composition of the central nervous system and that we designate as CNS. We have the capabilities to develop disease models through somatic cell reprogramming, iPSC sourcing and licensing, genome editing, as well as induction of disease models by chemical modifications. And we have well-established assays for impedance in order to assess cell survival and proliferation, multi-electrode arrays to assess electrophysiology, contractility assays in order to assess the cardiomyocyte functionality, calcium transient analysis, metabolic analysis, high-content imaging, gene expression, biomarker, and plate reader-based analysis. Examples from these assays will be presented as well. So I hope that in this brief introduction, I was able to highlight that Ncardia is ideally positioned to partner with clients for drug discovery projects, as we have the experience and expertise needed for iPSC sourcing and reprogramming, large-scale differentiation, assay development, as well as high-throughput screening. In the past few years, we have partnered with clients in order to study neurodegenerative diseases, cardiomyopathies and arrhythmias, oncology indications, musculoskeletal diseases such as Duchenne muscular dystrophy, as well as aging and senescence-associated disorders. We have worked with multiple therapeutic modalities, of course, small molecules, which constitute the majority of our projects, but also with more novel technologies such as AAV gene therapies, RNA therapeutics, antibodies, and antibody drug conjugates. With this brief introduction, I would like to pass on to my colleague, Ari, who will describe Ncardia's manufacturing process development and scale-up.
Thanks, Anna. Yeah, so I think it's a great pleasure for myself to introduce you and tell you a little bit more about the manufacturing capabilities that we have at Ncardia. And I think it's clear from Elena's introduction that one of the biggest assets of Ncardia is just the fact that we can start from iPSC generation and get it to the very end of using iPSC-derived cells in screening. And I think one very important component there is the ability to manufacture the cells. So in the next slide, I'm very happy to share the approach that we take, the lessons that we learned. For some cell types, I'd like to show some functional characteristics. And before we finish this session, Elena will close by demonstrating the use of these cells in drug discovery with some interesting case studies. So, as you're all probably aware of, is that iPSCs can be differentiated into theoretically every cell type of the body. On the other hand, it's very well known that iPSC biology is very complex. So these cells have to differentiate through different lineages, embryonic lineages, into the ultimate cell products. This is guided and regulated by intricate intracellular processes. We are very much experienced in the cardiovascular area and the neuronal area. We are developing processes in the immune area, and we are also active in process development for new cell types. But I'll not touch upon these activities in this presentation, but more or less focused on the established processes that we have in place. So just before moving into some more details about the cell types and the characteristics that we have, I thought it would be nice to share with you a little bit more about the approaches that we take, how we get to the high quality cell products that we need for discovery services. And this is just a quick summary of things that we learned over the last years. And what you can probably see is that we took into account lessons learned from pharmaceutical drug development, basically adapting the quality by design approaches that are so well known for drug discovery. And just to highlight a few very important things is knowing the complex biology and knowing that we want to get to really functional cells that can be of use in drug discovery. It's very important to define in an early phase what your target profile is. And this could comprise, for example, the identity that's needed for the discovery, the functionality that's needed, but probably even more important, if you have a high throughput screening in mind, the scale that we need to achieve. One of the things that we learned that it's very important that we established in-process analysis to make sure that we develop a process that's getting to the final functional cells that we need. Usually these differentiation processes from iPSCs to functional cells are complex, comprised of multiple steps. Generally, these are longer-term processes. So to really make sure that we're moving in the right direction, we need to understand where the cells are at every moment during that manufacturing process. And that's why, for example, for the cardiac and for endothelial cells, we have been really focusing on understanding what's going on there. You should think of analysis for identity using FACS. You could think about metabolites in the medium and all these kind of things. One focus that we had is to understand the critical process parameters, and I'll get back into that a little bit more detail. These process parameters are really determining the final outcome of your product, which we call CQAs, critical quality attributes, that we have been defined in the target product profile. And one very important thing is that we understand the relation between these two. So how do these parameters determine the in-process, but also the end-process quality attributes? And in the most ideal situation, and we probably need to now analyze multiple manufacturing runs, in the ideal situation, we understand the relation between these two so that we can come up with a control strategy to really improve the robustness of the manufacturing processes that we use to generate the cells for discovery services. One thing mentioned earlier, think about the scale that you need, in particular in case you want to move on with high throughput screening, and really in early phase move into the direction that you start using the right scaling technology. And I'll show you one or two examples in which we went up to the scale that you can achieve in suspension bioreactors, and that's actually the preferred way for us to get to those high scales that we need for high throughput screening. Now, altogether, hopefully, finally, I will be able to convince you that using this approach, we have been able to successfully scale up manufacturing of some iPSC-derived cell types. We were able to reduce the costs, and on top of that, we were able to improve product quality compared to processes that we used a decade ago. So, just quickly outlining the platform that we have been building in the manufacturing technology department. It's more or less a general representation. It's not that we are really kind of married to all these systems here, but just to give you an impression of what we use, we have from small scale to really large scale. There's medium equipment that we could use to generate larger amounts of media, and there's these high-end equipment that we can use to do in-process analysis, like automated flow cytometry, in-depth analysis of metabolites, high content imaging, and cell imaging as well. Now, to go a little bit back to the approach that I highlighted before, we have been developing processes, large-scale processes for iPSC cardiomyocytes, and I'll touch upon that in the next couple of slides as well. And the approach that we took is that we started from a small-scale bioreactor system, which allowed us to test multiple conditions, to optimize differentiation conditions, to optimize critical process parameters, like, for example, stirring speeds, which is of course particularly relevant for a stir tank bioreactor. Analyze whether we need a pH control, whether we need oxygen control, and so on. So that was all investigated experimental scale bioreactors. Now, as soon as the results were indicative of a functional cell types, we move into the next scale, which we call pilot scale in this case. And it was more or less used to really validate the defined critical process parameters, get more understanding of the process that we develop. On top of that, establish the relationships between them. There's options for process automation, also to avoid any human interference, which is generally well known for failures. And on top of that, with the aim to develop a control strategy. Now, we have been in this area for 1 to 2 years before we ended up here in the real large scale for IPC cardiomyocyte manufacture. Now, when I mention that we pay full attention to critical quality attributes and their relation with CPPs, this is not just something that you focus on during the development. This is particularly something that you continue focusing on when you go into commercial manufacture. So collecting more and more data to increase the understanding and to further improve the process to make it more and more robust. So after sharing some more detail about the approaches that we take to develop a process for large scale particularly, I'd like to share with you a little bit more characteristics of the cell processes and the cell types that we have in place at the moment. Of course, in this webinar, we are not able to touch upon every process that we have been establishing. So what we will do is focusing on iPSC cardiomyocytes, iPSC endothelial cells, and if time allows that, we would like to share some details about the iPSC cortical neurons. Of course, as mentioned by Elena before, there's more cell types that we have available for discovery services. And just in case you're interested, we're always happy to have another chat. And so please contact us for that if you want to know more about these cell types. So coming back to the iPSC cardiomyocytes, using that approach that I've just outlined and where we are right now in the large-scale bioreactors, we are able to, for reproducible manufacture of 2 to 4 billion assay-ready cells. So these are really the cells that come from a frozen vial that are adapted to adherent culture after thawing and that are ready to use in screening assays. On top of that, we have seen with our commercial cell line, we show manufacturing robustness in multiple batches. And we really see that this is making a huge difference compared to the processes that we used before based on adherent cultures. The success rate in the manufacturing that we do are over 95% with a particular commercial iPSC cell line that we offer. So that I really want to highlight that advantage of using controlled bioreactor systems as we do for iPSC cardiomyocytes. And on top of that, we have been working with many clients so far in which we use patient-derived or mutated, genetically modified iPSCs. And the process has been successful in the majority of the cases. Now, of course, knowing the complexity of iPSC biology, we also deal with iPSC lines that don't work immediately with the processes that we developed. But given the in-depth understanding that we have, what we learned over the last 3 years with our own iPSC lines and with additional client-derived iPSC lines, we are in a position to apply small process adaptations or fine mitigations to finally be able to deliver cells that are needed for discovery. Some data shown on this picture. What you can see here is just a reflection of what I just mentioned. So multiple batches, over 70% troponin T. This is all data coming from one iPSC line. So really proving that we have a robust process. The process also works for other lines like, for example, disease iPSC lines. On top of that, it's clear from patch clamp studies that the cell types that we generate with the process as we have it right now is really ventricular-like. Elena mentioned that we have processes available for atrial, but this profile really shows a ventricular-like behavior. A little bit more details. I guess we have a extremely low membrane resting potential there. And there's a good action potential amplitude there. There's an action potential duration of about 400 milliseconds. So a little bit more there based on immune fluorescence, a pronounced expression of presence of sarcomers that express myosin heavy chain 6, but also troponin T. and that's nicely associated with pelfactomy. One very important thing that I'd like to mention here, and I flagged that also very in a few earlier slides. So when we worked on scale up of the existing process that we had around 2018, we were really able to reduce the cost by 16-fold. And that was mainly due to three components there. So first of all, because we were able to develop a process that was much more efficient with a much higher success rate, we were able to reduce the materials that were needed for the amount of cells manufactured. We use bioreactors compared to using kind of outscaling T75 flask or hyper flasks in this situation. There's a real reduction on the labor that's needed, and a real reduction on the hands-on time that's needed for manufacturing of the batch. And of course, you know, there's also less space needed if you culture the cells in a bioreactor compared to multiple hyper flasks in an incubator. On top of that, and I realized this is data from 2019, which is already 2 years ago. The moment that we finished this process, we moved from 2 billion cardiomyocytes per FTE per year to the potential of 100 billion. I think we're at the moment we're even over that. Now, that's of course particularly relevant when you consider HTS, high throughput screening. It's even more relevant when you consider using these cells as in cell therapy. We're not going to touch upon that, but that's another focus point for Ncardia that we're currently building. We decided to include 4 additional slides, and there's of course much more to share about the functional characteristics of the cells that we generate. We just come up with some example data here using the adrenergic agonist isoproterenol. What you can clearly see is that the effects after treatment of the cells with isoproterenol are perfectly in line with the expectations. Both chronotropic and inotropic effects are shown, and the data shows increased beat rate as expected, increased spike amplitude. This increased beat rate is also associated with a shortening of the field potential duration, obviously. And on top of that, using contraction assays based on using impedance systems, we can see an increase of contractility if we treat the cells with isoproterenol, really confirming the inotropic effects that you would expect from such a molecule. Something of note may be relevant to mention, because there's always that debate about cells that you derive from iPSCs. Are they mature enough? At least what these results indicate is that the effect of isoproterenol on contractility is very much likely due to the expression of a receptor which is known to be highly expressed in adult cardiomyocytes, and that's data that we obtained from a RNA sequencing database that we have in place. Some other molecules that we test looking at other pathways. Mexiletine, a sodium channel blocker, nicely reduces contractility as well. Nifedipine dofetilide, looking at L-type calcium channels, looking at hERG channels, showing a profile that's expected. L-type channel, calcium channel blockers, reducing the field potential duration on the short-term treatment, but also on the longer-term treatment, and this is dependent on the concentration. On top of that, dofetilide, a well-known hERG channel blocker, prolongs the field potential duration after short-term treatment, after long-term treatment, and on top of that, we also observe the well-known arrhythmic effects that are known for hERG channel blocker dofetilide. So moving on with some more data, you may know that iPSC-derived cardiomyocytes are also used to assess safety of compounds that are used in the clinic. So what you're looking at here is profiles of cardiac function when treated with doxorubicin, ponatinib, tapsigargin, all cancer molecules that are used for cancer treatment, and there's definitely risks in using these molecules in patients. What we can see with all these molecules, actually, doxorubicin reducing the beat rate after 30 minutes treatment. There's not so much of an acute effect. Ponatinib, slightly different profile. There are acute effects on the beat rate. but definitely if there's a prolonged treatment there, and we see similar results, even severe effects of tepsigargine. And all these effects are considered to be deleterious for cardiac function. Another way to look at effects of compounds on cardiac function is looking at metabolism. We have different assays up and running. Probably I won't go into much detail there, but there's the oxygen consumption there. There's the Ncar that we analyze to analyze metabolic activity. Yes, so this is particularly reflecting cell stress here. This is oxidative phosphorylation that's being analyzed by the OCR. What you clearly can see is that doxorubicin has clear effect on the oxygen phosphorylation, is a clear inducer of cell stress. On top of that, there's a reduction of cell numbers in the cultures. Now, we decided to take along another molecule that has been tested in the clinic, but failed actually, has been withdrawn due to cardiac safety concerns. And very interestingly, there's not so much an effect on the cell viability there, on the number of cells. There's not so much effect on Ncar, but there's quite a significant effect on oxygen consumption rate. I'd like to move on with endothelial cells as another example that we developed a large-scale manufacturing process for. Just to introduce this a little bit better, it's well known that endothelial cells play an important role in human physiology, but also in human pathology. Of course, that's quite obvious because basically every tissue needs the blood vessels for supply of nutrients. So this is just listing a numerous diseases that has been reported to be related to cardiovascular functioning or dysfunctioning. I'd like also to touch upon side effects that have been seen when you use drugs. And I think a nice example has been published about antibody drug conjugates that are generally used as systemic treatments. So the vasculature will be in touch with these molecules, and it is known that these molecules can affect vascular integrity. In other words, lead to vascular leakage. So vascular leakage induced by drug treatment is known and could be of concern, a safety concern. Now, human umbilical vein endothelial cells, I would say, are the golden standard still in vascular research. But there's, of course, real limitations, and we have also been informed by our clients that these are limitations in pharmaceutical drug development. So there's batch-to-batch variation known for UVACs. By the way, that's the reason that many companies that supply those UVACs decide to pool multiple batches to just reduce the variation. But there's also risks in there that you dilute out the potential effect of your compound in the model. So there's heterogeneity, of course. It's hard to genetically modify these cells. And of course, as pointed out by Elena, that's the great potential of using iPSCs. So we decided to move into the area as well and develop manufacturing processes and assays for iPSC-derived endothelial cells to overcome these limitations. I'd like to be a little bit shorter here, just summarizing, and I'll show you some data on the angiogenesis assay that we developed. But in general, the cells that we manufacture, that we can manufacture at large scale, demonstrate a typical morphology of endothelial cells in culture. They express the right markers. They are not so much tissue specific at the moment, but I think everyone is aware that tissue specificity is more dictated by the environment and not so much by the genetics of the iPSCs. But at least they express CD31, they express CD124, which is a maturity marker. We have developed assays for those. If you want to know more about the barrier assay, I invite you to check Ncardia's website. And I'd like to show you some more background, some more details about the angiogenesis assay that we developed with these endothelial cells. It is clear, and we have done project with clients, it's clear that these cells can be used for compound testing on an endothelial barrier on angiogenesis and also on cytotoxicity. We're not going to show any more details about these two in this presentation. It's also clear that they can be effectively used in discovery, efficacy, but also safety related to drug-induced drug-induced vascular leakage. This is just representing a model of the assay that we developed. We basically use a fluidic device comprised of multiple channels, 3 channels in total. So there is one channel in which we place the endothelial cells. There's another channel which we do the angiogenic factors. So there's a gradient developing there which attract these endothelial cells, actually activate them to sprout and form a new vessel. Of course, this is all model, and I'm happy to share real results with you. But finally, we use high-content imaging for data analysis. And there's quite some publications out there that you can check if you want to learn more. And of course, we can arrange separate conversations about that too. So the data here, already after 24 hours, you can see clear sprouting, and this is really evading after 24 hours. If you look after 7 days, there's a real capillary network formed there with branches. And what is even more interesting, at least in my opinion, that there's a lumen formed. So there's a fluorescent tracer injected here, and that tracer leaks to the other channel because there's a lumen formed by endothelial cells. You can also see that these capillaries are leak tight, relatively leak tight. Of course, the goal that we have is to use these models for drug discovery. So I'd like to show you an example in which we tested a model compound, sunitinib, which is a known angiogenesis inhibitor. So we tested the UVEX next to the iPSC endothelial cells that we manufactured. And what you can clearly see is that there is dose-dependent effect of sunitinib. Another thing that's very important to note is that this is happening at pharmacological levels, physiological levels. We have been developing algorithms to quantify this here. There's different ways that you can look at this, the distance of migration, the capillary area, of course, that there's a number of nuclei that you can measure. And based on some assay analysis, we finally decided to focus on migration distance because that was really distinguishing treatments from control. If you look at the signal window, which is relatively high, there's a very good C prime factor there showing that the assay is quite robust. And there's good percentage CV there, at least acceptable if you compare with reference values. And this is another quantification of that. The concentration range of sunitinib really reduces the formation of those sprouts. We didn't only test sunitinib, we also took another angiogenesis inhibitor, more inhibiting metabolism in cells, 3PO, showing similar results, but not shown in this picture here. If you're interested, this data has been published. Okay, so the final cell type I'd like to touch upon is cortical neurons. In the past, we developed processes to manufacture cortical neurons. We actually took it a step further and made a catalog product out of this. But as you can imagine, for this presentation, it's very relevant to understand how we could use these cells in discovery service projects. Before doing so, I'd like to show you some characteristics of these cells. It's known that cortical neurons, they are part of course, of the central nervous system, and they are really responsible for associative and cognitive functions. On the other hand, it's well known that they play a vital role in many neurodegenerative diseases, such as Alzheimer's disease and Parkinson's disease. We developed a process resulting in postmitotic, mainly glutamatergic, cortical layer committed neurons. One criterion that we believe is really important before we release these cells for use in assays, they should express more than 80% of FOX3G1, which is a marker of cortical neurons. On top of that, there should be more than 50% expression of CPTIP2, which is a marker for cortical neurons, particularly in layer 5. But there's of course more characterization that we do. We also check TBR1, and you can see the stainings in this presentation here. We check beta-3 tubulin, which is a really general neuronal marker. So these cells, before we use them in projects, they're highly qualified. So there's a couple of more slides in which I would like to show you some characteristics of these cells, and also share with you how we start developing readouts and assays for use of these cells in disease models. But of course, there's also a white paper associated to this webinar that you're welcome to read. There's other material out there that we indicate how we could use these cells in Alzheimer's disease, Parkinson's disease modeling. So one of the things that we did is really optimize the culture of these cells to get these particular profiles of neuronal activity. I'd like to go over these slides a little bit faster because I would like to give Elena the chance to touch upon the case studies that we have in place. Of course, the cells show neuronal activity, firing activity, but what is very important to note here is that we do a co-culture with Ncardia astrocytes. We really see emerging stable networks of bursts, kind of indicated here by the pink squares. I'll go over these next slides relatively quickly. This webinar will be shared, so everyone will be able to have a closer look at this. We did additional assays there, looking at calcium imaging, testing model compounds, really see that GABA agonists can really reduce the signals, the firing signals in these cells. On top of that, we have been setting up model systems to look at lysosomal dysfunction, mitochondrial dysfunction, really dysfunctions going on in neurodegenerative diseases. We have been focusing on testing model compounds, setting up high content imaging algorithms for proper readout. And the goal is now to implement these models in disease modeling and discovery in the neuronal field. One additional piece of data here using rotenone to model neurite outgrowth dysfunction. You can see those neurites here. They can be quantified using our algorithms developed using the high content imaging system, and there's a clear effect of the model compound rotenone there. And as mentioned, we very much look forward to implement those model systems in neuronal drug discovery. So I'd like to refer to the case study that we have been presenting on the website that we have available attached to this webinar in a data container. So I'd like to pass on quickly to Elena to allow you to tell us more about the case studies that you prepared.
Thank you, Ari. Really great examples of manufacturing processes and assays developed. And we'll pass on to describing briefly how some of these assays have been used in our projects. The first example that I will touch upon comes from a Friedreich's ataxia disease model, and here iPSC-derived cardiomyocytes were manufactured at large scale from wild-type iPSC lines as well as cell lines that were patient-derived and carrying Friedreich's ataxia-associated mutations. These cardiomyocytes were initially characterized at the basal level in order to demonstrate that they did have reduced protein levels for frataxin protein, and then assay development was performed in order to identify cellular phenotypes that showed a good distinction in functionality between the wild type and the patient-derived mutated cells. Using optimal assays, and in this case, we selected the study of reactive oxygen species levels to assess oxidative stress, as well as calcium imaging analysis to assess calcium handling capabilities, were reversed using a gene therapy candidate vector. Therefore, this iPSC disease model was very valuable in assessing the efficacy of early therapeutic candidates. The second example is from a stage 1 high throughput screening campaign that was performed in order to identify small molecules that could be repurposed for the use in hypertrophic cardiomyopathy indications. Initially, iPSC cardiomyocytes were again manufactured using a wild type and mutated iPSC lines, and an assay was developed measuring the clinically relevant marker BNP in order to determine differences between wild type and hypertrophic cells. In parallel, we developed high-throughput screening protocols in our TCAN Fluent automation platform that would allow us to screen libraries of approximately 3,600 small molecules. From this primary screen, 341 hits were initially identified, and 192 hits were confirmed using further secondary screening that included dose response analysis, exclusion of false positive hits, and orthogonal assays. With further safety assays, we were able to narrow down the potential candidates for drug repurposing to 25. So this was a very helpful and successful high-throughput screening campaign using a clinically relevant biomarker in an iPSC-based model of cardiomyopathy. The third case study will jump into the field of neuroinflammation, and in this case, we utilized our inside astrocyte cells, and we produced the chemically induced disease model by treating the astrocytes with a cocktail of cytokines that induced an inflammatory response detected with IL-6 secretion, which is a known marker for neuroinflammation. This study is ongoing, and it intends to screen over 100 compounds in order to identify which derivatives of the lead compounds are most efficacious in reducing a pro-inflammatory response in these iPSC-derived astrocyte cells. And the final case study that we will present today is one that was related to cardiotoxicity assessment. In this case, anti-cancer therapeutics were developed and were efficacious based on in vivo studies against oncology indications. However, they were associated also with bradycardic effects, especially with atrial fibrillation and atrial arrhythmia side effects. So we used our iPSC-derived atrial cardiomyocytes that were manufactured in really large scale in order to develop a custom multi-electrode array-based assay that would allow us to detect these bradycardic effects and then perform structure-activity relationship studies in order to identify derivatives of the small molecules that would be both efficacious but also safe for clinical use. So with this information, I hope that we have indicated that Ncardia can partner with you with our 10 years of experience in order to offer end-to-end services in iPSC-based drug discovery for cardiovascular, neural, and neural indications, but also any other indications that may be of interest to you. We offer services that span iPSC reprogramming, genome editing, and cell sourcing, large-scale manufacturing for several cell types, as Ari described in detail. And we have already developed a number of functional assays that include contractility, calcium analysis, biomarker analysis, metabolic analysis, and so on, that we have used in successful projects. So this brings us to the end of today's webinars. I hope that we have offered a good understanding of how iPSCs can add value to the drug discovery pipeline and what considerations need to be taken into account when designing a project. And of course, that partnering with experts such as Ncardia can help overcome these challenges. Thank you for your attention, and we can open the floor to any questions that you may have.
Yes, thank you very much, both Ari and Eleanor. And just a quick reminder to the audience, in order to ask questions, you can type them into the box at the top right-hand corner of your screen and click submit. But in the meantime, we have some questions coming in. So the first question to both Ari and Elena. One person is interested in the HIPSC-based drug screening for Alzheimer's diseases. So are you able to help?
Yeah, I can take this one. Absolutely, we're able to help. So to establish an Alzheimer's disease model, we would most likely source iPSC lines that carry MAPT mutations or any other mutation of interest, and then manufacture, for example, cortical neurons that are able to manifest the disease phenotypes. Most often in Alzheimer's disease models, it's necessary to provide a seed of aggregated proteins, and this can be in the form of either human recombinant tau protein fibrils or insoluble extracts isolated from brain tissues from patients suffering with frontotemporal dementia, for example. And these are needed in order to exacerbate the disease phenotype. After setting up this platform, we can perform assays such as looking at phosphorylated tau protein aggregation, neurite outgrowth, mitochondrial and lysosomal trafficking, and stress, and other similar assays that can help us elucidate good assays, good phenotypic readouts for Alzheimer's disease.
Sure. Thank you very much for that, Eleanor. So another person says that I would like to use your motor neurons for their drug screening. Do you, in this case, you could say manufacture this type of cell?
Yes, I'm happy to take this question. Thanks for this question. Indeed, I outlined the established processes that we have, and I decided to focus a little bit more on how we got to these established processes. So in case you would need motor neurons or, for example, any other cell type of the body, actually two things that we could do. First of all, we could check whether these cells are available and whether we can source them from another supplier. That's one thing. On the other hand, and we are working with clients at the moment in that direction, we can apply our capabilities and knowledge and expertise in iPSC manufacture to develop new processes. So of course that's different. That's a different type of project. We are moving into that direction with some clients, and we will potentially be able to do that for motor neurons as well.
Sure. Thank you very much for that, Ari. Another person asks, can you perform a screen using siRNAs of gnRNAs for target identification?
Yeah, that's a great question. So we can definitely perform those kinds of screens, and we can either receive libraries from clients or purchase them directly from vendors, and set up the necessary transfection protocols to perform screens with siRNAs or guide RNAs. And of course, there is an additional benefit to performing screening campaigns at Ncardia, which is that our facilities are set up in a sterile environment, so that the long protocols required for iPSC cultures can all be carried out with no problems of contamination. And this is unique compared to other screening facilities where cell cultures are maybe only 1 to 2 days long, and this doesn't require such high standards of sterility.
Sure. Thank you very much for that, Alina. So in this case, how do you account for the immaturity of HIPSC derivatives then?
Yeah, that's a great question, and one that we take into consideration for every study. And typically we address this by enabling enough time in culture for the cells to acquire the right characteristics and gene expression profiles, so that they can give us physiologically relevant responses to known pharmacology. And then we know that the appropriate ion channels and structural proteins and so on are expressed.
Sure. So, Ben, between both you two, are you, in this case, are your established manufacturing processes easily applicable to new patient lines?
great question again. Happy to take on this. I touched upon this relatively quickly in the webinar. We established processes for Ncardia commercially available iPSC lines. We know that these processes can be transferred and used for external iPSC lines coming from patients generated by introducing a mutation. On the other hand, of course, we're also facing situations in which we work with iPSC lines that don't immediately work with the processes that we developed. What we have seen in some projects is that we went back to the data that we applied, the knowledge that we obtained when we developed the processes. Then we introduced some small process modifications. So that's one way to finally get to the cells that we need for the project. And the other options that we have in-house, and that's the way we really work, and has been appreciated by clients. There's always mitigations that we may consider, like moving into different systems, scaling out shaker-based systems, for example. I think, the message is we know the goal of the client, and our goal is to deliver the cells that are needed for the project.
Sure, thank you very much for that, Ari. So there's quite a long question coming up here next. So one person asks, but how old are the neurons showing activity in the MEA assay in co-culture with astrocytes? And do you also see electrophysiological activity in cultures without, in this case, astrocytes? And if so, at which time of neuronal or differentiation?
Okay, that's a great question. So the presence of astrocytes facilitates the electrophysiological activity, especially because if populations consist of purely glutamatergic cortical neurons, that creates excitotoxicity and the cells suffer. Whereas if astrocytes are present in the culture, this reduces the glutamate levels and improves the activity. In terms of the time in culture, typically we do wait, we do see electrical activity very early after thawing cells, maybe from 4 to 7 days after thawing, but we really do wait until 21 or 28 days before we consider the cells ready for functional characterization.
Sure, thank you very much for that, Alina. And the final question, one person does ask, do you harvest HIP-SC from placental cord blood?
Yeah, thank you for that question. We don't have that process established. Nevertheless, if cells were, if you had access to cells that were derived from placental cord blood with the appropriate documentation, then we would be happy to take on the cells from you from the reprogramming stage.
Mm-hmm, sure. Ari, anything else to add on to that before we conclude today?
No, thank you. I have nothing to add there. It was all clear from Elena's answer. Thanks.
No problem, that's fine. And thank you very much to the audience. Just a quick reminder, if you did not get your question answered, they can be answered at a later date. So that just leaves me to thank both Ari and Elena for what a great presentation, and to Ncardia for sponsoring this event. That's all the attendees. You'll receive an email shortly on how you can access the on-demand version, or you can access it at our website, www.business-review-webinars.com. We look forward to sharing future webinars with you, so please do keep an eye out on our website as mentioned, and follow us on Twitter at BRWebinars for daily updates, and join our LinkedIn community group. Once again, thank you all very much for attending today's webinar session with Ncardia, and hope you all have a very great day. Thank you for attending.